Hello, everybody. Thank you for joining us on this webinar, Introduction to In Vitro Permeation Testing, IVPT, Strengths and Weaknesses. My name is Bruna Lousada, and I am the European Sales and Marketing Manager of Teledyne Hanson. And this webinar will be presented by Clive from Toxicology and Frank Toner from Charles River, and sponsored by Teledyne Hanson. Before we get started, let's take care of some housekeeping items. Although we are on mute, you can submit your questions anytime by using the Q&A section on the bottom of your screen. And we will do our very best at the conclusion of this presentation to answer all of your questions. If we run out of time, we will email to you directly. If you're having any trouble hearing, or if there are any connection problems, we suggest that you log off and log back on. You should also know that this presentation will be available for replay on the Teledyne Hanson YouTube channel. Okay, so let's start. Teledyne Hanson is an innovator and premium manufacturer of dissolution and diffusion testing instruments based in Los Angeles, California, in the United States. In addition to the products and service that we offer, Teledyne Hanson is committed to supporting the global pharmaceutical industry with educational material such as this one. So please check the Teledyne Hanson website and our social media channels often so you don't miss any of this great content. It is my pleasure today to present our speakers. So the first speaker is Clive Roper, and he's the director of Roper Toxicology Consulting Limited. Roper Toxicology was born out of 30 years of in vitro toxicology experience, developing new tests and new approach methodologies and bringing this to market. They can take your ideas to regulatory agencies, CROs, and global pharmaceutical, cosmetic, and agrochemical companies. And they specialize in topical product development and delivery strategies for new product development. The next speaker is Frank Toner, and he's the Associate Director of In Vitro Dermal Services at Charles River Laboratories. Led by internationally recognized experts, Charles River team has been running into in vitro skin penetration studies for decades, delivering the data that drives decisions and helps gain regulatory approval for agrochemical, pharmaceutical, and consumer products. So with that, I'll turn it over to Clive and Frank.
Thank you very much, Luna, for the very kind introduction. I'll just give a short disclaimer. The opinions expressed in this presentation are those of the authors and do not represent the opinions, official policy, or position of Roper Toxicology Consulting, Charles River Laboratory, or any client, customer, agency, or employer. So we've got a number of little topics that we would like to discuss with you today. So we're going to introduce you to IVPT. So the first question, of course, is what is IVPT? Why do we perform IVPT? A little bit about the draft guidance on Acyclovir, a summary of the draft guidance on Acyclovir. Strengths of the draft guidance, there are always strengths of guidances, but we also have identified some important weaknesses which we will be open to discussing with you. There's kind of 5 steps in this guidance, so we want to provide some suggestions on how to strengthen the test. Please, it's really important, please discuss with the FDA before initiating your test. It's something that's so important that we've had to decided that we wanted to incorporate this into the introduction. Discuss with the agency if you are providing new ways of working. They're very, very engaging and they're very interested. And of course, if they don't like your ideas, it's better to go ahead without. But it's better not to go ahead with the test. There's going to be some more information for you to follow up. And we will then be very open to talking with you with your questions and answers. And as said, please put them in the chat box, your questions. So of course, the very first question is, what is IVPT? So IVPT stands for In Vitro Permeation Testing. IVPT uses vertical diffusion cell systems to detect an API in receptor cell medium at various time points. And here's some good examples of materials that have been through this type of test. The goal of IVPT is to assess the performance of a formulation matrix. That could be, for example, a cream or a gel. It's really up to you what to test, but we need to be very thoughtful as to the appropriateness of that choice, and as to whether that would be something that the FDA would choose to be a relevant material. But we're looking in terms of its ability to permeate certain organic barriers, such as the skin, but we could also be thinking about other tissues. The flux profile is measured and graphed, and the IVPT results are expected to correlate with in vivo behavior. So it's quite important to realize that this is a replacement of the clinical trial, so which is very interesting to be performing, and very interesting that regulatory agencies find this important. Why do we perform the IVPT testing? Well, in vitro permeation testing is performed for safety assessment. It can be used in lead candidate and formulation selection and prediction of clinical data. In this case, what we're really focusing on is bioequivalence testing for the FDA. But there are different ways that we can perform IVPT. And this is an example of some work that Frank and I did a few years ago, where we were looking at the bioequivalence of two formulations containing butanethine hydrochloride. Now, we really recommend that you have a look at this paper because it involved some very simple process of generating bioanalysis data, then testing very similarly to we would do in the FDA guidance to check that we could actually see differences. And then we did some basic statistics comparing the two formulations. Now, this was accepted by the FDA, but as we all know, there are different offices in the FDA, and this study would, although it was accepted, would not be suitable for to be accepted by the Office of Generics. But it's very useful to read to see how this is done in reality, with some changes in statistics and study design. Another question is why perform in these IVP tests? And this is another study, again, performed by Frank with Bayer. And the in vitro and in vivo comparison of this drug, BAY10038003, was performed in vitro in skin absorption tests and in vivo in clinical trials. Now, the first step was required to compare the ointment, the hydrophilic cream, lipophilic cream, and milk. And with all of the data provided, they were able to choose which two formulations they wanted to go ahead with in the main clinical trials. And actually, when they performed these, they predicted very nicely the results that were seen in the clinical trials. Again, please do read this. It's got all the details for you. Now, what we're going to focus on is the generic topical clinical trial. So as I said before, the purpose here is to replace a clinical trial for generic topical formulations. It is really important that the drug penetrates through the skin, because what we're measuring is absorbed material, the material which reaches the receptor fluid in our in vitro studies. The receptor fluid represents the systemic absorption, and this is what we analyze. We then measure and calculate the flux and area under curve, and then we compare these statistically between the test and the RLD, the ready marketed formulation. The test and RLD formulations are compared for bioequivalence, and they can be modified with discussions with the FDA. And again, really recommend talking to them. They're really engaging. They're really open to the ideas. And as I've said before, the test is a new or generic drug formulation, and RLD is the marketed or reference listed. So we do have draft guidance, and this has been performed on Acyclovir. And again, it's very clear that it's an in vivo clinical trial is your first option. It's not for this presentation. It then moves on to the IVRT, and if you can remember a few months back, we had the fabulous presentation, Introduction to IVRT, by Sasha Gorinson and Michael Herbig. And again, do recommend that you go and watch that. It's a great presentation. Now, it's not a clear and straightforward guidance because you've got these different options and a lot of information. So we're going to try and talk you through. And now I'm going to move across to Frank to take you through the strengths of the draft guidance.
Thanks, Clive. So looking at the draft guidance for Acyclovir, the things it does describe are the conditions and quality processes that must be followed. It clearly documents the equipment that must be used and the parameters of those equipment. It has good information on the test system itself. It talks in detail about the method validation and references both the IVPT method validation and the associated bioanalytical validation requirements. The receptor fluid and the suitability of the receptor fluid requirements are discussed. And then the statistical requirements and the statistical design are clearly defined. So some weaknesses or ambiguities in the draft guidance. How the overall testing strategy is divided up is open to interpretation. And as Clive mentioned earlier, what do we do if the drug does not penetrate or Jmax is not observed? Because these topical products are designed to be active in the skin or on the skin, and therefore absorption is expected to be limited. And just as an aside, whilst this is a replacement and an alternative to a clinical trial, it doesn't replace the clinical trial in its application procedures. So we are not expecting from this guideline to follow the application amounts that would be done clinically or the duration of exposure and multiple exposures. So the 5 steps to follow in a bioequivalence package. So here are the 5 steps that we would follow in our laboratory when performing these packages for our clients. So first of all, from the analytical method development, and that would be a non-GLP study in our laboratory, and then following on from developing the analytical method, you would then validate it in accordance with GLP, following the appropriate bioanalytical guidance documents. Then moving on to the actual IVPT testing itself. First of all, conduct the method development studies, and they are not required to be performed GLP and are not performed GLP in our laboratory. And then on to the more defined pilot and pivotal studies that are performed to GLP in our laboratory. So I'll take you through points 1 to 5 in a bit more detail now. So that analytical method development. So the purpose of it, obviously, is to develop the method for receptor fluid analysis. So key aspect for this is the analytical sensitivity and ensuring that we have suitable analytical sensitivity, because with that limited absorption for some APIs, we need to ensure that we do not get below limit of quantification for our receptor fluid samples when we come to the IVPT testing. And it also has the suitability of the receptor fluid for that API itself. for stability. And then once we've developed that method, we then validate that method in accordance with GLP and the appropriate bioanalytical documentation. Normally, for these APIs, it would be by LCMS-MS. It confirms the suitability of the method and then creates our analytical method that we follow for the IVPT studies. So moving on to step 3 is our IVPT method development. So there are many aspects of this that are clearly defined in the draft document for Acyclovir. So first of all, we have the IVPT apparatus qualification itself. And the apparatus that is used, either vertical static cells or flow, as a minimum, must have the exposure area or the area of the orifice confirmed, and that where the receptor chamber, where the receptor fluid is collected, the volume of that must be known and determined, and then that control is in place to maintain the skin surface at 32 plus or minus 1 degree, which leads us into the membrane qualification for the skin itself. So we have the confirming the barrier integrity, the stratum corneum barrier integrity, and that can be performed by 3 suitable methods, being tritiated water absorption, transepidermal water loss, or electrical resistance stroke impedance. And the qualification must be done for every time it is used to confirm that barrier integrity, and then that is referenced against historic empirical data to confirm suitable barrier integrity. And then that membrane, as I said, must be 32 degrees plus or minus 1, and that's confirmed prior to application of any formulation. And then also for the duration of any study, an undosed control sample per piece of apparatus would have periodic temperature monitoring to confirm that that temperature is maintained over the duration of the study. Then looking at the receptor fluid solution underneath the skin, that is qualified in the analytical method development. And the receptor solution makeup, it must be a physiological based receptor fluid of suitable pH and containing an antimicrobial to stop any growth within that receptor fluid at those elevated temperatures of 32 degrees. And it must not be rate limiting for the absorption of the test item. So you must determine suitable solubility of your API in that receptor solution. And then when we come to sampling that receptor solution, again, that must be qualified and data provided to show that that is both accurate and precise. And if a vertical static cell is used, then it will contain a magnetic stirring bar, and you must confirm that the stirring speed is adequate and continuous, or for a flow cell that the flow rate is suitable for collection of the material. And then the receptor solution undergoes the analytical method validation to show that it is robust and acceptable for the API of choice. For example, for stability, both at that elevated stability over the duration of sampling at 32 degrees, or during storage at ambient or frozen prior to analysis. And the laboratory that you perform the IVPT study in must be environmentally controlled, and the temperature of the laboratory must be maintained. It's recommended to be maintained at 21 plus or minus 2 degrees centigrade, with a relative humidity of 50% plus or minus 20% as a range. So moving on, again, ambiguities or things to consider in the draft guidance for Acyclovir. The statement, the study duration should be sufficient to characterize the cutaneous pharmacokinetics of the drug. And that leads into including sufficiently complete flux profile to identify the maximum flux and a decline in flux thereafter across multiple subsequent time points. So the question is, what happens when or if those are not met due to the limited absorption of a specific API? And for that, again, as Clive said, there are a number of things that can be done, and the FDA are open to novel solutions, and there are some that are mentioned in the guidance, such as removal of the formulation during the duration of the study, or potentially looking at the pharmacokinetics in the skin itself, but these would all be discussed with the FDA prior to working. But ideally, the aim of the IVPT method development and associated analytical method development is to gain suitable sensitivity, so you get at least 8 non-zero sampling time points, and that you see that maximum flux with a decline afterwards. And that is the aim of some of the earlier IVPT method development studies. And then moving on to the next part of the IVPT method development. So as we touched on earlier, just things to consider, that the draft guidance doesn't relate to how the many topical drugs penetrate the skin, as many don't penetrate the skin as they're active on or in the skin. And it should just be noted that the guidance doesn't help comparing efficacy in or on the skin, as we're looking at that receptor fluid. However, it is useful for that transdermal or possibly systemic risk assessment data. And then other aspects of the IVPT method development. So on our discussion with the FDA, we've concluded we need the following aspects. So we perform these studies for 96 hours. And the purpose we do that for that period of time is it answers the question of, well, what if we ran it longer? Would we see that flux, peak flux, and then decrease? And that is the longest we would recommend running it for in our laboratory. During that time, we take frequent receptor fluid collections. And when we are making any comparison, either between dose weights or different formulation types, we use a minimum of 4 donors from 4 replicates. Obviously, as Clive will mention, for the Pivotal study later on, the number of donors for that study may be greater based on statistical comparison. And for analyzing sensitivity, that is where we discriminate between different application rates or dose amounts. We'd recommend testing 5, 10, and 30 as the starting points. The guidance does mention 5 to 15 milligrams per centimeter squared, but also says that you can go outside of that area if required. And this has later been clarified in the FDA workshops. And again, just going back to potential options or simplifications that could be done but are not in the guidance. So it could follow the actual clinical application. So the amount that we applied clinically, it could be a single or multiple dose at that fixed application, and for mimicking the duration of a clinical dosing. However, the guidance is clear that by confirming sensitivity by different dose weights and selectivity by the alternative that Clive will talk a bit more about in the pilot study. This gives the confidence of the robustness of the IVPT methodology so that the statistical analysis of bioequivalence is strengthened. And then moving on to the last part to consider for IVPT method development, it's just why simplify? What would be the options in simplifying or benefits? So in clinical use, a topical product is washed off by a patient in everyday life. So we could incorporate a wash off, and we would follow the actual patient's use. So one of the great strengths of IVPT study is being able to mimic that clinical or in-use scenario by having the same amounts, by following the same procedure that would be done clinically. And that would allow us to identify the true differences, and we could confirm the RLD test and alternative fulfilling the success criteria. Okay, and now I'll pass back to Clive.
So step 4 is the IVPT pilot. Now the pilot is defined by the preliminary IVPT method development. The objective is to identify the number of donors and replicates for the pivotal test and confirm the selectivity of the test. So the selectivity being that we can see there is true differences. There's no consideration of the intradonor or interdonor variability. So this can be dealt with balancing test groups by barrier function. We are often only evaluating the skin and not the formulation. We have seen some examples where it's so affected by the barrier function, and yet we're asked to randomly select the groupings. So a barrier function test and rank order could be used with the highest to lowest. For example, we could do a test being dosed with donor 1, cell 1 and 6, donor 2, 3 and 4, donor 3, 2 and 5. The RLD could be then 2 and 5, 1 and 6, 3 and 4, and the different 3 and 4, 2 and 5, 1 and 6, for example. So this would mean that we're using 9 donors, 2 replicates per formulation per donor gives us 54 samples. So it's not a small test. What we are probably now doing is not having these insane level of randomness. So it's really important to think about the different levels of barrier function that we're seeing within the same donor out across different donors. And we are seeing some real problems when we're doing this entirely random. So the IVPT pilot continued. We run the test. We confirm the alternative formulation is statistically different. We perform the statistics and we choose the comparators. So we have area under curve and J max. And we can also look at the total absorption at specified time points. So this then allows us to identify the number of donors, and we have 4 replicates per donor. And each time a formulation is applied to a replicate, it is still only 1 person. So even though we've got 4 replicates per donor, which might be 2 replicates for each formulation, we've still got 1 person. So 1 replicate from 10 donors would be 10 samples, 20 replicates from 10 donors, and still n equals 10, still 10 donors. So is it more appropriate to increase donor number than replicate per donor number? Well, this is another discussion point, but you have to follow the statistics, which does take all of this into account, this intra and inter-individual variability. There's not actually much discussed on the pivotal because you've effectively come to the point. You're really rerunning the previous study, but with much greater numbers of replicates that have been told that you have generated by the previous study. So the IVPT has the same considerations as the pilot study. The statistics are well defined. We use the same comparators from the pilot, AUC and Jmax, or total absorption at time points. And this is, remember, this is considered to be a clinical bioequivalence test, so the same conditions of statistics are applied. So you're doing the same statistical analyses if you were doing this in the in vitro model or the in vivo model. We do have more information that we refer you to. There's the draft guidance on Acyclovir with all of the details, the M10 bioanalytical method validation draft guidance, an introduction to IVRT webinar video, which we introduced before, the in vitro data, bioequivalence data for topical product bioequivalence review perspective FDA public workshop, and the IVRT and IVPT workshop from last summer. So we thank you all for joining us, and we are now open to answering your questions.
That's great. Thank you so much, Clive and Frank, and well done. This was a great presentation. I'm sure it was very interesting to our audience. So as Clive said, we are now open for questions. So just a reminder for everyone, so you can submit your questions using the Q&A button at the bottom of your screen. Okay, thank you. Let's start. Okay, so here's the first question for Clive and Frank. So is there a true correlation of IVPT studies to clinical performance? I mean, is it possible to generate an in vitro, in vivo correlation?
I've given some answers there. It's Clive speaking. I've given some answers onto these. I hope you can still read them. When we do IVIVC, of course, we need to make sure that we are doing like for like. So yes, if we're doing a clinical performance where the skin of the human patient is normal, and very frequently, of course, with topical drugs, it's not, then we will get a good correlation. If the skin is diseased, then we won't get a good correlation because these are intact human skin donors. Of course, the old way that we used to do IVIVC, we failed as well because we used rats or animals or pigs to compare against clinical patients. So there's a nice paper that Frank has got with Bayer, and I've put the DOI number there in the chat. And they basically used some work that Frank did to predict the right formulations to take into a clinical trial. And when they compared the two clinical trial formulations with the 2 of 4 chosen in vitro, they were able to show that it was quite predictive, and they got a good result from that. Certainly when we're looking across into any other area, outside pharmaceuticals, then these tests are used in safety assessment, and again, there's some work that demonstrates that this is protective and predictive of the human worker or consumer exposures, but that's a different story. Frank, did you want to add to that?
No, I think you covered that very well, Clive. Thanks.
Thank you, Clive. Next question, can you please comment on the difference between the FDA and EMA requirements?
I've again answered some of these, and the differences are big. You would not be able to use both an FDA and an EMA bioequivalence study. The FDA is very precise about what you must do. So they probably would not accept an EMA design. I think you would probably have better chance of getting an EMA design, an FDA design through an EMA, but again, I think this would be very difficult. The requirements are certainly different, and not just even the way, I mean, even the way statistics is done, it's actually the way the studies are performed and the way that they're set up, and even the way that they describe them. So, but both of them are draft guidances when I last looked. So certainly we would be happy to discuss with people more about the differences, but probably well beyond this lecture today.
Okay, thank you. From your comment on slide 17, IVPT is not really suitable testing method for drugs that act on or within the skin, right? If meaningful concentrations does not permeate into receptor medium, meaning the systemic exposure is minimal.
I've answered, but I'll let Frank have a go this time.
I think it was Frank, yeah.
Yeah, so IVPT studies in general are useful because you can analyze what's in the skin as well, as we talked about. This specific guidance has tried to go for a simple approach in that if we can have the suitable sensitivity in the receptor fluid, and that is in the receptor solution underneath the skin, and you can tell the differences between different dose rates and an alternatively different formulation, then by that we've demonstrated suitability of it for the guidance. But other approaches are to do a mass balance study or a time course study where you actually look at the amounts in the different layers of skin, and that's fairly common. So looking at the amounts in the stratum corneum layers and the epidermis and the dermis, as well as that receptor fluid. Clive, you got anything else to add there?
No, I think you've got it, and I've made some comments as well. I hope you can all read it too.
Thank you, Frank. Next question. How do you handle when unconventional flux profile is observed or no Jmax, even with 96 hours duration?
Frank, do you want to answer? I've answered again on here, but
yeah,
do you want to answer? Yeah.
So as Clive said in his comment, that under the classical, under the guidance, the Acyclovir guidance, it wouldn't be a suitable candidate because you need those two parameters for your statistical comparison. And this is where we're talking about there may be where we look at more innovative ideas. So that example that is discussed in the guidance of removing the dose partway through the duration, so you see a decrease after that. So that would allow a decrease in flux after it. That needs careful consideration in the receptor fluid development phase, because we need to accurately and reproducibly remove the amount so that we're not introducing an artifact. And that would definitely be one that, as Clive says, that would be you'd want to discuss with the FDA if we're taking a different approach from the standard guidance document.
Okay, thank you, Frank. I don't think we have any other questions at the moment. So I think that's all for today. Clive and Frank, thank you so much. And of course, thank to all of you, our participants, and especially the ones who asked questions. Those were really great questions. As I mentioned earlier, this webinar will be on the Teledyne Hanson YouTube and social media and website within a few days, and you can watch this on demand. Thank you very much.